All right, this is Anatomy Physiology lesson 10.07 over the sodium potassium pump. And what we had said in the previous videos was that the sodium potassium pump is a membrane protein. And this is not an example of passive transport. This is an example of active transport. So it's a membrane protein that operates via active transport. Which means that it goes from, now you'll notice here it says extracellular space. This means this is outside the cell. And intracellular space means this is inside the cell. And what's going to happen is we're going to take something that is not equally concentrated, and we're going to actually make it more concentrated. So what we'll see is we start off and we have a little bit of sodium outside the cell. By the time we're done, we're going to have a lot of sodium outside the cell. So we go against the concentration gradient. And we're not going to use this. You can kind of ignore this. Really not going to I don't want to confuse you with the sodium and the potassium here, so I'm going to use my own thing. So we're not going to deal with that. But what we have here are 4 different instances. This is the pump at rest, and then we start, and this is the process by which the sodium potassium pump creates a charge difference between the outside of the cell and the inside of the cell. So what happens is, using this active transport, the whole goal of this protein is to create a charge difference, on the opposite sides of the membrane. Of the cell membrane. And in fact, when we're done, what we're going to see is the sodium potassium pump creates, if we put a probe here, a little voltmeter. So remember, voltage is a measure of electric, in a sense, of electric potential energy. So V for voltmeter. And if I put one probe here, and I put one probe here, and I could see what my voltages are, this would be 0, and this would be negative 70 millivolts. So we actually, and you'll see this on batteries. So one of the things I forgot to mention when we were talking about voltage is that's how batteries operate. If you put a battery in your smoke alarm, that's a 9-volt battery. That allows you to move charges with that 9 volts of potential electrical energy. A AA is 1.5 volts of voltage. Your neurons, and this is the cell membrane of an axon of a neuron. This little, and not fancy membrane protein creates a charge. So that's its whole goal, is to create that charge difference. So before we go too far down this rabbit hole, I want to review ATP. So down here, I have a molecule of ATP, and that stands for adenosine triphosphate. And if that sounds familiar, you remember adenine, guanine, thymine, cytosine from nucleic acids from DNA? Your body actually takes adenine and it kind of it retrofits it. It slaps on some phosphate groups. So each of these little, in mine it's blue, each of these represent a phosphate group. So this is the adenosine sugar, and you've got phosphate, phosphate, phosphate. So that's what the triphosphate is. We've got adenosine triphosphate. And I'll redraw it here. And I'm going to do it with, put some distance in between. So the energy, and this is a P, and this is the adenosine group. The energy in adenosine triphosphates lies in the bonds. especially this first bond, the bond that I put the arrow to. Now there's a little bit of energy in this, some energy in this bond. There's a little bit of energy in this bond, not much, but this, tons of energy. And so what will happen is our body systems will take ATP and it'll crack off this phosphate group and we'll get ADP. So over here, here's my adenosine sugar. Those are my two phosphates. So diphosphate instead of triphosphate. Well, where'd the third phosphate go? It's up here, and we're going to talk about why, what's going on with that. So, and you'll notice here we've got a little bit of a firecracker. That's showing that when that broke, it released energy. So, and not just a little bit, but a lot, relatively. Okay, so that's your crash course review of ATP. So let's talk about what we have going on in our diagram now. The first thing you need to know is that the sodium ions are the hexagons. So Na, all of these are Na. And the ovals are potassium. So these are also transporting ions, just like our membrane ion channels, but they're doing it in a different way. They're doing it via active transport and ATP, and they're going against the concentration gradient. So remember, this is against the concentration gradient. Against the concentration gradient. So when we get started, let's kind of do switch colors here. Let's go with step 1. And I'm going to go to green. So step 1, the protein, we'll do letters here. The protein is at rest. And open to the inside of the cell. That's what we have here, intracellular space. So this guy's sitting here and it's open, and it has 3 sites that can bind. It has 3 open sites for sodium ions to bind. So this third one's empty. We have 1 bound, 2 bound, the third one's bound. This is outside the cell, this is inside the cell. And what we see happening right now is we see the sodium entering into the membrane protein, into the sodium potassium pump, and binding to those sites. So what we actually have happening here is sodium ions are binding. That's what's actually going on in step 1. And I'm going to kind of, I'll outline that in green. All right, in step 2, do purple. So in step 2, in purple, the first thing that happens is here comes this ATP, and the ATP is going to bind to the protein. So the ATP binds to the pump protein. And we go from here to here. So we've had a couple of things that happen here. This bang represents the breaking of the phosphate-phosphate bond. So the phosphate-phosphate bond in ATP breaks. And we said that releases a lot of energy. And the protein uses that energy to change its shape. So the next thing that happens is the energy release changes the shape of the protein and it causes it to open up to the outside of the cell. So here we were open to the inside, but we're using that energy change to open to the outside. To the outside of the cell. Once that sodium leaves. So this actually shows about 3 different things happening. So letter D, 3 sodium ions leave LEAVE, leave the protein to the outside of the cell. Okay, the next one in orange. So here goes my ADP. My ADP's leaving. It's taken off. My protein's open to the outside, and if you see there's a little arrow here, it shows something going into the pump, but now these are potassium ions. So for frame 3, the ADP leaves, and there's binding sites for 2 potassium ions. So we get 2 potassium ions, enter and bind. And if you notice here, we still have this phosphate group that's attached to the protein, whereas over here we don't. So what causes it to go to flip from the outside to the inside is the phosphate group leaves. The phosphate group leaves. Causing the pump to open to the inside of the cell. So here we are then, we're basically back at the beginning. So we're going to repeat this process. So for frame 4, I think pink will show up. Not very well. Let me pick a different color here. Let's try light blue. Light blue. Yeah, that looks better. So our process will repeat. And what we're doing is we're sending 3 positive out and 2 positive ions in. You should know if you do that. Think about if you're walking up a staircase. If you walk up 3 steps and then you walk back 2, you walk up 3 and you walk back 2, you're actually going to get to the top of the staircase. You're making progress forward. So as we do this with every cycle, we have a net +1 out of the cell. And so we're building up sodium ions. Remember, they're, let me do them in orange. My color coding the same. So we're getting hexagons here. I'm just going to switch to circles because it's taking too long. So I get all of these sodium ions built up outside the cell. That means that I'm going to have a positive charge here. Well, you might be saying, wait, what's this -70 about? Because you said I'm going to have a net -70. Well, voltage, when you measure voltage, is a little bit funny because it matters where you put the probe, whether it's positive or negative. If I put my 0 probe here, this would say 0 and this would say +70. But because I put the 0 probe here, in a sense, it's 0 and -70. If you think about it, this is still more positive than this. Even though this is a negative number, if you compare it to this is more positive. So the end result is that the whole purpose, this causes a positive voltage outside the cell. and negative inside. And what we're going to see now that we've tackled all of the review material is that this voltage difference is what allows the nerve impulse to spread down a neuron. If it wasn't for the sodium potassium pump, you would not have the ability to transport nerve impulses. Not only that, but creating differences in charge is also how cellular respiration works. So if you remember from biology, we talked about the fact that we build up electrons outside and then we open up the electron pump and the electrons flow through and they cause stuff to happen. So creating that charge difference is what really is so much of your body is all about. In this case, we're doing ions, sodium and potassium. Other places we'll do electrons, some places we do chloride ions. But to recap, oops. So the whole point of the sodium potassium pump is we're using active transport, which means we're using ATP to form a concentration that isn't there. And in this case, the concentration is sodium ions outside the cell, gives us this voltage across the cell. So we have steps, and yes, you're going to need to know all of these steps. You've got the protein at rest, and we get the sodium coming in, the ATP binds, back to phosphate, opening it to the outside, sodium released. Phosphate group hangs around while the ADP leaves, the potassium come in and bind, and then when that phosphate group leaves, which we see over here, it opens it back up to the beginning. And once we hit this -70, this process kind of stops. So this gets it to, it's kind of like your thermostat. It gets the temperature up, and then when the temperature's at the point at once, it all stops. That was your overview on sodium potassium pump. We're ready to start talking about action potential in the next video.